Hello everyone, welcome to GDC classes English. So today we have our next topic for the MCQ discussion based out of biopharmaceutics on the absorption of drug. By far we have already covered one absorption of drug part. So this is going to be the second part. And again I am telling you this is going to be useful for anyone who is preparing for any government pharmacist exam or even GPAT, NIPER, DI, whatever you are preparing for. This is going to be one sufficient MCQ attempts video with the explanation. So make sure to mention your scores in the comment section below so that even we can assess how much you have secured, as well as any feedbacks or improvements or even any suggestions are most welcome. So make sure you put them in the comment section below. Okay, so let's start with our first question. So first question says endocytosis is called as corpuscular vesicular transport, both A and B, or convective transport. Now endocytosis means engulfing, right? We have different methods of mechanisms of drug absorption starting from the paracellular, transcellular, and this one includes the vesicular transport. Now this vesicular transport is based on basically, for example, the process by which amoeba obtains food, if you all remember, that is termed as endocytosis. So the process of engulfing, so the corpuscles are actually formed. Now they engulf the material, the drug, and then transport it across the membrane. Basically the oily or high molecular weight molecules are generally transported through this mechanism, right? So it is including both corpuscular as well as vesicular transport. Okay. Next, which of the following is the other name of cell eating? So we have seen endocytosis, which is the engulfing. Endocytosis is a part of phagocytosis. So generally, broadly, if we classify, we have phagocytosis and we have pinocytosis. Phagocytosis means cell eating process. Pinocytosis means cell drinking process, right? So phagocytosis mainly involves the cell eating process. For example, where does this happen? You have seen the examples of natural killer cells, macrophages, which eat up the your toxin or any other bacteria. They try to engulf and destroy it by using the phagocytosis process. Okay, next. What influences the permeation of drugs in an ionic or electrochemical diffusion? So once the ionic or electrochemical diffusion is mentioned, although the diffusion is primarily based on the concentration gradient, but here it says ionic or electrochemical, which justifies that we are talking about none other than the charge based, right? We are not talking about only the concentration gradient because it is influenced by the ionic or electrochemical diffusion. It will be based on the charge on the membrane. Otherwise, the simple or passive diffusion is basically concerned on the electrochemical gradient. Okay, next. Transfer of endocytic vesicle from one extracellular component to another component is known as what? Is known as transfer. Basically from one side using the endocytic vesicle to other side, that will be known as transcytosis. We have seen the phagocytosis, pinocytosis, endocytosis. So transfer using these vesicular phenomena, that will be known as the transcytosis. Okay. Direct questions that are asked are basically directly on the definition. Here I think yes, so yes, the process of movement of unchanged drug from the site of administration to the site of measurement, that is known as, now you all have seen the answer because of the slide, but it is okay. This is a direct definition based out of the drug absorption process, which basically includes the movement of drug from the site of administration to the systemic circulation, right? So this is known as the drug absorption process. Okay, next. Which of the following is not an advantage of a rapid absorption pattern of a drug? Not an advantage. Okay, right. So first, lesser susceptibility of the drug degradation. This is correct. If the drug is being absorbed quickly, then there are chances that from the bloodstream it has started to absorb, so there will be much lesser chances of the degradation process. Higher blood levels and rapid onset of action. Obviously, the absorption is being quicker, so the onset of action will also be quicker, right? More uniform, greater and reproducible therapeutic response. This is also fine because the absorption has been rapid. There will be a uniform absorption, so as the rapid onset of action as well as the therapeutic response will be fine. So lower blood levels and slower onset of action. So this would be the correct answer because they have asked the which of the following is not an advantage. So we know option D is not an advantage. Okay. Next. Which of the following is not a theory of drug dissolution? So in the drug dissolution process, we mainly focus on three theories. Number one, which is the simplest and most important, which is the basically the first, I think the layer model, right? So it is the diffusion layer model in which we assume two layers. One is going to be the stagnant layer, other one is going to be the diffuse layer, right? Second we have is the interfacial barrier model. Third we have is the surface renewal theory. So we have three theories: diffusion layer model, penetration or the surface renewal theory, or the interfacial barrier model. So fixed law of diffusion, this is not a part of the drug dissolution process. It's a part of the diffusion, passive diffusion process. Okay. So we have diffusion layer model or film theory. We have Danquart model or surface renewal or penetration theory, interfacial model or the double barrier or limited solvation theory. Okay. Next question. A process in which a solid substance solubilizes in a given solvent. Now from the name it suggests process in which a given substance is solubilizing. That means solubility is enhanced or the dissolution is happening. So is it absorption? No, we have seen the definition. Deaggregation? No, there is no breaking. Disintegration? Not at all. And then we are led to the dissolution process. Direct definition based question? No. Extra concept here is required. So dissolution is a process in which solid substance solubilizes in a given solvent. Now this is explained much better with these three theories which you have named. Very important: diffusion layer model, film theory; Danquart surface model or penetration theory; third, interfacial barrier or limited solvation theory. All these three at least names you must remember. Which of the following is a rate determining step of the drug absorption when the drug is given orally? Okay. Now, disintegration and deaggregation, are these any which way rate limiting step? Not at all. Disintegration and dissolution, dissolution and permeation through the membrane, and the disintegration. See, disintegration is never a rate limiting step, right? Disintegration is never a rate limiting step, but dissolution can be a rate limiting step if the drug does not have a good solubility profile. Second, we talk about the permeability. Now, if the drug is having poor permeability or poor ability to cross the membrane because of any reasons, not a proper partition coefficient or anyway higher molecular weight, then the permeability is going to be the rate limiting step. So for the rate limiting step, we consider dissolution and the permeability profile. Option C here it is going to be correct. Okay. Next. Which of the following is true about the pore transport? Now, pore transport is also known as the convective transport, which focuses and works on the principle of the solvent drag, where it is created due to hydrostatic pressure. The drugs are actually transported, which are hydrophilic in nature. They are transported with the solvent drag. Water flux is created and the drug is transported. So, pore transport is primarily used for the absorption of the large lipophilic molecules. I have just said it is for the hydrophilic molecules, which are very small in size. The driving force will be the osmotic pressure, hydrostatic pressure, leading to the movement of water along with the small molecule. Now, this matches exactly my current description. Pore transport is ineffective for water-soluble drug. No, it is effective for the water-soluble drugs. Chain-like compounds with molecular weight above 400, not at all. It should be less than 100 Dalton. only then as well as hydrophilic to be able to pass through the pore transport, right? So we have option B as our correct answer. Next, which of the following route has rapid absorption from the aqueous solution? Number one is the intravenous, intramuscular, subcutaneous, or both B and C, right? So we have rapid absorption from the aqueous solution. Now this suggests that the rapid absorption will take place from both intramuscular as well as subcutaneous routes. Okay, so correct answer will be both B and C. Let's see how. So in the intravenous there is 100% complete systemic drug absorption. Why? Because the drug is directly available in the bloodstream. For the intramuscular injection, there is a rapid absorption from the aqueous solution, although it will turn out to be slow absorption for the non-aqueous ones. In the subcutaneous also there is a rapid absorption from the aqueous solution and slow absorption from the depot formulations. Okay, next, which of the following is generally used for vaccines and drugs which are not absorbed orally? So for vaccines and for drug, either we use intravenous route, intramuscular route, subcutaneous route, or even the buccal or sublingual. We all know that these vaccines are usually administered by the subcutaneous route, right? Subcutaneous injection. Okay. Next, match the column with the particular description. Right here we have routes. Okay. Now buccal, sublingual, oral, rectal, intravenous. Okay. We all know when the drug is administered through intravenous route, that means the bioavailability is going to be 100%. That means the fraction which is available, that will be available, like for the 100% it is going to be available. So for the fourth we have Q. So we have our two options available, right? These two are wrong. We have our two options available. Okay. Now for the rectal part. For the rectal part, we already know that it will cause a discomfort condition to the patients. So we know that the answer now is going to be the option number C. Okay. Still we have the buccal route for where we ensure that there is no pre-systemic circulation or metabolism, or the first pass effect is much hampered. Okay. Then for the oral, that is suitable for both immediate and modified release products. Even if you know basics of the routes, it is very easy to answer. So option C here it is correct. Okay. Now moving on, which of the following which part of the membrane is responsible for the relative impermeability of the polar molecule in and out of the cell? Now in and out of the cell, that is dependent upon how the polar head and the non-polar tail part is actually placed, because the membrane is made up of the biphospholipid layer, right? Membrane is made up of the biphospholipid layer. So basically the molecules which are much more lipophilic in nature, they are welcome and are very much transported, right? So which part of the membrane is responsible for the impermeability of the polar molecule? So if the molecule is going to be polar or hydrophilic in nature, then the hydrophobic core is going to stop it from the transportation. Otherwise, if it was hydrophobic or lipophilic, it is very much welcomed. So option C will be correct. Okay. Next, the applicability of Noyes-Whitney equation describes which order of kinetics? So this basically the Noyes-Whitney equation is used for the dissolution rate explanation, describes the rate of drug dissolution in context to the surface area, partition coefficient, concentration in stagnant and the bulk layer, as well as the volume. So here it is said that it will definitely focus on the first, dissolution rate. Okay. Okay, so this is a simple example. You can spot the equation right here. This is the DAK, which is the partition coefficient, into CS minus CB, which is the concentration in stagnant, minus bulk. A was the surface area, D was the dissolution coefficient, upon VH. V will be the volume and H will be the thickness of the layer that is formed. Next, the size of the aqueous filled pore is how much angstrom? Now this size is actually focusing on the membrane. So you know the composition of membrane, which is a biphospholipid layer, right? The membrane is going to be the phospholipid layer, right? So in this case, we generally say the thickness is around 25 angstrom, but we have a very limited hydrophilic space or the aqueous channel in between, which have a size range from 4 to 10 angstrom, right? So the limited hydrophilic space is around 4 to 10 angstrom, which is filled by the aqueous pores. Other than that, the thickness of membrane is going to be the 25 angstrom. Okay, clear? So this is the arrangement of the membrane. We have proteins which can be extrinsic, we have proteins which can be intrinsic, and we have the lipids in which we have polar head part shifting to outside and non-polar tail part inside. Appearance is somewhat of a sandwich like model, and then we have our what we say aqueous pores, as the size is 4 to 10 angstrom and the thickness is going to be the 25 angstrom. Okay, now which of the following is a pseudopolymorph? Hydrate, solvate, both, and none of these. Now this question is basically a part of the pharmaceutics, not necessarily the biopharmaceutics, but before we judge it because of the dissolution process that can be changed by the change of the polymorph as well, right? So which of the following is a pseudopolymorph? Both these hydrates and solvates are categorized as the pseudopolymorph, right? So they the anhydrous form has greater aqueous solubility than the hydrates, right? When the solvent is water, then it is hydrate. Other than water, it will be the solvent. Next, the total body water volume for a 72 kg person is, now it's from a table in which we have seen the markers and the physiological compartments as well as the volume of each physiological compartment. So they're specifically asking for a total body water volume. Now this is, I'm telling you, the table I'll show you now is very important. So this is going to be the 42 liters. Notate down. The plasma compartment or the vascular compartment will be volume will be 6 and the plasma will be 3 liters. The ECF, which excludes the plasma, will be around 12 liters. The ICF, which is the intracellular fluid, which is around 24 liters, and the total body water will be around 42 liter. Okay, next, which of the following is useful in the estimation of rate of absorption? Is it Cmax? Is it Tmax? Is it AUC? Or is it MEC, minimum effective concentration? For estimating the rate of drug absorption, we generally have the Tmax, right? We generally use the Tmax. So these are all the pharmacokinetic parameters. We have peak plasma concentration as Cmax, right? We have the time of peak plasma concentration, which is the Tmax, and the area under the curve, which gives the rate as well as extent of drug absorption. So here we have our last question. Which is the another name for active transport? Downhill, uphill, passive, or bulk flow? See, bulk flow is the other name of the convective or pore transport in which the hydrostatic pressure or water flux was created, and due to the solvent drag, the drug molecules were transported. Then we have passive diffusion in which there is no energy is required. Just on the basis of concentration gradient, the molecules are transported. But here it is a downhill transport. Passive transport is always a downhill transport because it does not require the expenditure of energy. But the active transport requires energy, and thus we call it as a uphill transport. Okay, so another name will be the uphill transport, right? Here you can see the summary in which all the transports can be seen. We have number 1, passive transport, just the concentration higher to lower and no use of energy. Then primary active transport from lower to higher, that means against the concentration gradient, that will require the use of energy. Now energy in the form of ATP. This is a primary transport, or we also call it as a uniport, because it is transporting single molecules in single direction. Then we have secondary transport. You can see it will be symport if both the molecules are transported in the same direction, or antiport if the both molecules are transported in the opposite direction. But these both require the use of energy, so we call it as a uphill transport. So that's it for today. I hope the questions were fine to you all. You can mention in the comments below, as I said in the start of the video, and we are really looking forward to your responses. So that's it for today. We will meet in the next video. Thank you so much.